All right, here we are. Welcome, everybody. Thank you so very much for joining us for another online event from the Fleet Science Center. This one is our Sharp Mind series, which we normally host on Monday mornings in our Highcroft Dome Theater at the Fleet Science Center. But as you know, COVID struck and we find ourselves at home. But really happy to be able to still offer you science engagement through virtual programming. And we're very excited today to be working again with the Sanford, Burnham, Prebys Medical Discovery Institute. They've been a longtime partner of the Fleet Science Center. They do some amazing research, some amazing work helping people with diseases. And we're excited to have another speaker joining us for that tonight. A couple of things about the flow tonight. You are in a Zoom webinar, so this is not your regular Zoom room. If you have a question, please use the Q&A function. There is a little speech bubble symbol at the bottom of your screen that you can use, or the top, depending on where your control panel is located. Just hit that, type in your question, and we'll get to that at the end of the presentation by Dr. Hansen. You can type the question anytime. You don't have to wait until the Q&A. So if a question pops into your mind, I would suggest just writing it down, typing it in that Q&A. Then we make sure that we get to it, and you don't forget and we don't forget. The Fleet was excited to open on the 1st of July. Unfortunately, now with new restrictions, we have to close again tomorrow. So if you were hoping to visit us, we're really, really sorry. If all goes well, we'll be back in 3 weeks. Until then, I do hope you'll join us for many more talks. We have a talk on mass coming up next month for Sharp Minds. We have forensic science coming up. We have gene editing and gene splicing talk coming out. So there's lots of exciting things you can still explore with us and keep learning, keep staying curious, and keep having some fun with science. Our amazing speaker today is Dr. Malene Hansen. She's a fantastic researcher and also one of our two scientists walk into a bar scientists. So when we can all go back to bars safely, and you come across the two scientists walk into a bar program, then you might be able to talk to her face to face over a glass of beer. Sounds pretty exciting in these times, doesn't it? Malene, why don't you take it away?
All right. I share the screen first, or?
Yes, go ahead.
Here we go. So let's see if it all works. I hope you can see it.
Yep.
All right. Well, it's a pleasure to be here. Thank you, Andrea, for inviting me and also for helping facilitate the partnership with SVP where we work. I'm excited to tell you some of our work at the Institute today. I thought I would just let everybody know who we are and where we are. This is a shot over the mesa. So SVP, let me get a pointer here. SVP is all these buildings here across the North Torrey Pines Road where the Scripps hospitals are, and UCSD out here in the back. We're a nonprofit research institute, like Andrea mentioned. It's about almost 700 of us, about 40 or so lab like ours, and we do both a lot of fundamental science, but also discovery, drug discovery to look into different human diseases. And so there's a lot of cool things going on, along with also training the next generation of scientists, something that I'm also very much involved in aside from the lab job of doing research. So that's what I'm going to tell you about today. And I'm mostly going to tell you about work that we do with our favorite organism, which is a small, tiny earthworm called C. elegans. And I will tell you mostly about trends that we've learned from such simpler organisms in terms of how they use cellular recycling to live long and healthier lives. So we don't know if these ideas are yet to hold true in humans, but they look very promising, as what I'm going to show you is at least true for worms and flies and mice. And so possibly it can be something that can be translated one day also to humans. So aging is something we're all familiar with, including myself. You can see here a picture of myself as a very young scientist, almost 30 years ago at something called Novo Nordisk. It's a big biotech in Denmark that makes insulin. I started doing research there. I didn't really know at the time that I wanted to study aging. Obviously, I was maybe even way too young to sort of think that way. And this is what I do today. What I did at the time, not at Novo Nordisk, but when I was doing my PhD at Copenhagen University, was to study cancer biology. And so you may know, and this is kind of what got me into aging, is that cancer, along with a number of other very serious diseases, increases or is not only a common, but also the biggest risk factor for these diseases. So for cancer here in red, you can see you're at higher risk to get cancer when you grow older. This is also true for other diseases such as respiratory diseases like pneumonia. So this would be relevant in these times of COVID. You've probably also heard that older people are at higher risk for something like COVID. So this is sort of the hard facts, including also when you hit around your 50s, you can see you get a much higher probability of having a serious what's called neurodegenerative disease like Alzheimer's. So this is sort of a fascinating kind of idea that there are links like that. The other thing that fascinated me and still does is that there are some people that seem to be living much longer and essentially disease-free lives. And this would be sort of the personal best for humans. This is Jean Calment. She lived in France to be 122 and is the person, at least on the books, that so far has lived the longest life. And this is, I guess, despite her not so healthy life habits. But what really came sort of from all of this is this idea that, and that really fascinated me to be this researcher I am today, and that is how does aging happen at the molecular level? Like we can see aging and these links at sort of grant levels for people and at broader levels, but what is it that actually happens inside our cells of the body as time passes by? This is not only something that we do. There are many research labs, both here in the US and worldwide, that are looking into understanding these so-called molecular or genetic mechanisms that may underlie aging. And the ultimate goal in this regard would obviously be to learn something about aging that, as a common denominator, if you wish, could help us fight against not only one, but many age-related diseases, like I showed you on the slide before. So to sort of go this kind of in level of detail, let me just remind you how this kind of works at the more, cellular and molecular level. So you would all know that the cells of our bodies consist of a number of cells in different organs. Each cell has a nucleus and also other components, which I will come back to. But the nucleus is key in that it holds chromosomes, which are basically the storage of, or is like yarn that is like DNA, and the DNA stores basically information. So think of it as DNA encoding like a particular gene X. That's like a chapter in a book for a particular protein with a function. And so think of, for instance, enzymes in our guts that are produced in our pancreas that needs to help digest our food. That would be their function. So the equivalent, like I said, maybe you can think of DNA as a book that holds all the chapters with all the information, each of one, to make a certain protein. You can think of as a tool, and the tool has a function, again, like an enzyme, or it could be more of a structural role, like the proteins in our nails, for instance. So there are many, many of those, these proteins with function, and they are key for us to basically work and be healthy in a cell that's normal. However, damage can happen, and cells would basically over time accumulate such damage. You would well know that this is something like we all meet in our day, like UV light, I'm sure heard, can help damage the DNA, and this would create problems in terms of over time, you could see different types of damage. And many of these have been sort of dubbed hallmarks of aging. And so I'll just give you a few of the ideas as to what can happen that's sort of bad to a cell. So I already mentioned to you DNA or damage to DNA can, of course, cause problems to the book. You can't really read the chapter well anymore. There are particular problems at the ends of these chromosomes. These are called telomeres, and they tend to shorten in some cases, and that obviously would be a bad idea as well. You lose, again, information in this book of tool making. Genes, as of the products from the DNA I mentioned, can be altered over time, not do their normal thing. Protein can be damaged as well, again, if they're not made the correct way. There are ways that also cells communicate with each other. Obviously, one cell is not alone. There are many cells in a body, and they will talk to each other. So as you would also see examples of, there are sort of receptors or little antennas that can sit on the cell surface that receives information from other cells, and that can also kind of go sort of a little haywire. And then another major one that I will talk a little more about is that mitochondria, these are these powerhouses of our cells that can generate energy for the rest of the cell to function normally, can also kind of get damaged and start leaking basically signals that also impact these other components. And overall, it leads to aberrant function, like I already mentioned. In some cases, actually, it can also lead to basically secretion of what we would consider inflammatory molecules, of bad molecules that, again, can cause bad effects to the next door neighbor that may not itself have experienced this very same damage, but can be affected by other cells that have. So this is all dire news. That's obviously not so good, but luckily, and probably for good reasons, our cells have figured out ways to try and repair this. So, that's sort of the good news, that there are ways that the cell has to, and genes and other processes that work in them that I will talk about, that sort of can counterbalance the damage that would happen over time. And so that's obviously where this whole idea also to help prevent aging, or maybe even one day reverse it, or at least help us better understand some of these age-related diseases, is if we could learn more about these genes and repair processes that can sort of help set the balance right, or tip it in a way that could maybe be helpful for us in the long run. So that became sort of the quest almost 30 years ago, really for real, to look for so-called longevity genes using these very simple model organisms. And here comes the worm that I mentioned already as our favorite model organism. And it has a lot of advantages. It's very tiny, by the way. This is my hair in a microscope, and you can see these are 5 little worms that are by them. So they're kind of 1 millimeter long. They live in normal soil. If you go out and take a teaspoon, there will be hundreds of them in there. They normally eat bacteria in the wild. That's what we feed them as well. But for the purpose of really using them for our research, they're really great for a number of reasons. First of all, they do not live for a very long time. They live for a couple of weeks. However, at the same time, they still have high physiological complexity, like they have neurons and muscle and skin, just like we have. So we can think of ways to at least parallel many of the things that we see. And then, of course, what we don't do in humans, we don't do experiments to that end, but we can do many types of experiments in these worms in terms of turning these genes on and off to see what happens with the animal. But really the key part is that worms, like I said, age too, and they actually age very fast over a few weeks. And as you can probably appreciate here, actually we show this, we host a lot of high school students, and they always get this right. They can always tell an old worm from a young worm. They kind of look wrinkled, even if you look at a very high power magnification scope, and they tend to stop moving. I'll show you a little movie now on how they tend to age. They get infections more like humans. So there's at least a number of ways we can think of that they have some traits that are similar to how we age. So the question really began in that way to ask which genes could affect aging and lifespan, again, taking full advantage of the fact that aging itself goes very fast and is kind of accelerated in this organism. So back to the little scheme I used to, that I introduced to you before, right? This idea that DNA has information about genes, proteins, and functions. As a geneticist, what you would do is you would on purpose try and mutate this DNA, like kind of in a directed way, not just by shining UV light on it, sort of in an undirected way, but we have ways to actually introduce mutations in a directed way to now mutate the DNA that gives rise to a different looking protein that also would then, and this would be the question for us, how would that affect the function of the animal? In this case, looking at the function that's aging. So that was done a long time ago. Some of the first screens was done by the professor I trained in the lab with. Her name is Cynthia Kenyon. She was a professor at UCSF. She's now head of Calico, a Google-based longevity company up in the Bay Area. But what she and others did in the late 80s and then beginning of the 90s, almost 30 years ago now, was to look for basically gene mutations that would create animals that, like here in red, created animals that were longer lived. And so what I'll just show you here is a little lifespan curve. This shows the fraction of worms alive over time. And of course, the key here is that this is days of adulthood. For a population of worms, we would usually look at about 100 or so. We would look at them every other day. Some are not so lucky, they die early, some die a little later, and some actually live quite long. So you get sort of this span, which by the way would look similar if you look at the human population. Some would die younger, some would die older, but we all call it like we have a mean lifespan of a certain amount that would be around 76 years, for instance, for humans. So that's wild type animals. But then what was really cool, and the seminal experiment that was done back then, including with work from Tom Johnson's lab, was to show that a gene mutation in a single gene actually caused these animals to, first of all, die later, and also at a, as you can see, a slower pace. And these animals, importantly, actually looked healthier for longer, which of course was a very important objective for something like looking at the longevity mutant. So I'll show you a movie that Dr. Kenyon shared with me. It first shows a normal worm. It's basically moving around in its food. It's swimming in its sort of little layer of bacteria. You can see it moves happily. This is a mutant worm at the same sort of younger age. This is kind of at the edge of the food in this case, but you can see it looks very similar to the wild type animal. It has rigor and it searches well for its food. It will have progeny, but here's a later time point and old adults, basically very old adults now, a few weeks old. You can kind of get the feeling it doesn't move much. You can almost see it's kind of become flaccid, and it has sort of almost these moon craters of wrinkles literally in its skin. It is more susceptible to infections. Here's another shot. You can see this guy not moving very much. This is probably already dead. And so this is at the very curve of its lifespan. Now here instead is at the very same old age, these mutants that I showed you. So you can see these animals look much healthier. They have nice muscle tone. They still search for food and eat, and are basically healthier. And also, and I'm not showing you this, but they're also less susceptible to some of the diseases that we actually can mimic in the worm. I won't really tell you much about today. So these are healthier animals, and obviously the question became, so why do they live longer? What's sort of wrong with them? They had a mutated gene, as I mentioned, and the gene that was mutated in this particular worm, in this DAF2 gene, is a receptor that's actually turned out to be very interesting because it was similar to a receptor or a protein that we all have, and that's the human insulin receptor. So here's a real recap of that how that works. A receptor, like I said, is a protein that would sit here on the surface of the cell, receiving signals from the outside, in this case from insulin, which is a hormone that basically floats around in our blood, in particular when there's a lot of food available. So every time we take in a meal, insulin will tell the cells there's lots of food available. You can increase your metabolism, store goodies for later challenges, and that would be sort of a healthy thing. However, the mutant, like I said, actually has a less active insulin receptor. It's not gone, it's just not as well functioning, so it doesn't really sense insulin very well. So that means that the cell itself actually thinks that there's little food, even though it's maybe swimming in food. And that in itself, and this is sort of key to the concept of many of these longevity mechanisms, is that it decreases metabolism and activates what the cell has as these protective mechanisms. And I will tell you more about that in a second. But overall, that ends up delaying aging, at least in worms. And so that was originally the case, like found many years ago, like I mentioned, but later work actually in flies, these live a little bit longer, like the case of months. And then also mice, as you know, that is a common use model organism. They live even longer, like a couple of years. But what was common for all of them and us is that we share this insulin receptor. And at least for these three different and very distal types of animals, they all link to aging. So if you reduce the receptor a little bit, like we did in the worms, they would all enjoy a longer and healthier lifespan. So that's very exciting, of course. It doesn't prove in any way that this is going to hold true in humans, but I'm just going to show you this little picture to highlight that there are very long-lived humans. They live to be more than 100. They're called centenarians. And it's proven to be the case, at least I think as far as the 6 group or 7 groups that's been looked at. Obviously, we don't do experiments in terms of turning gene on and off, but you can look at their genes and sequence their genome. And actually, it turns out that they have a higher chance of having mutations in this very same gene and in other genes that basically work with this insulin receptor. So that doesn't prove that this is why they live long, but it does raise the question, the very interesting question, whether it's possible that the IGF-1 receptor could affect also longevity in humans. So this is still to be proven. In the meantime, and this is a busy slide, what I just wanted to say here is not only this insulin signaling, but also many other, and what's common for them, are nutrient sensing genes and processes that have also been linked to aging in a conserved fashion, so meaning in multiple different organisms. And so what I just wanted to highlight with the nutrient sensing part is, of course, this point that we would all be thinking about. So is there a way that we could maybe possibly engage these processes in a way that could be beneficial? And so you may ask yourself, so how can we do that? If you don't want to mess with the genes, which obviously is not an ethical thing, and we don't do that in humans, but you can think of other ways that we could maybe modify genes like that. And so one piece of news to that end is something called dietary restriction. And you may all have just eaten, which is always the case. But this is actually something that works very well even in monkeys. And this is one of the latest experiments that has been done. So to the left are monkeys that have been kept in captivity. They live for about 30 years. So you can imagine this is a very long-lived and very long experiments compared to the simpler organism I already mentioned to you. But the animals on this, on the right side, were kept, in the same facility, but they were fed a little less food. And the key here is a little less, because obviously if you give them too little, it's effectively starvation. So dietary restriction by definition is reduced food intake without malnutrition. So if you do this, and this doesn't only work in monkeys, although this is where it's been tested, and probably the most complex organism and primate it's been tested, can extend lifespan in a wide variety of animals, including these monkeys. So obviously the question is, so how do we do that? Because personally, I love to eat. So are there other things that we could maybe do that would not necessarily involve eating less? And so the good news is that there are actually some chemicals and some drugs that can also do the part about affecting these nutrient sensing processes that I already mentioned. So these are either natural compounds or engineered drugs. They have been tested extensively, again, in these simpler organisms, the worm, the fly, and the mouse. Resveratrol, you may have heard about. It's to be found in red wine, not really in very high concentration, but that's where it is. Rapamycin, it's an FDA-approved drug, actually against immunosuppressants for transplants or organ transplant. And then there's metformin, as you may also know, millions of people are taking this as an anti-diabetic compound. And what's been found, and consistently so, is that these compounds, at least in these organisms, seems to extensively prolong lifespan and also make these animals healthier for longer. I want to emphasize that none of this has been tested thoroughly in humans. There are some clinical trials that are underway for testing some of these, including metformin and something in a study called TAME. So I want to caution that, again, there's no good news yet for humans, but certainly for the worms and the flies and the mice in the audience. The question, of course, is, at least from our perspective as geneticists interested in understanding aging, is how do these drugs do this? How do they affect these nutrient sensing pathway? And that brings me to the sort of key part of my talk and the specific topic of research for our lab, namely the cellular recycle as one of these molecular mechanisms of aging. So what's true not only in the insulin mutant I already mentioned to you, and for dietary restriction, there's a number of other ways actually that animals like worms, but also flies and mice can be made longer lived. I won't tell you all about them here. Suffice to say that for all of these long-lived mutants that we have found and we study extensively in these simple model organisms, is that they all extend lifespan, at least in part, by engaging this cellular recycling. And so I want to tell you a little more about that. A few years ago, you may have heard that this process, also called autophagy for self-digestion, basically, that's what it literally means in Greek, was awarded to Professor Yoshimori Osumi for basically deciphering this process. And I'll try and explain that to you now. I assume that you're all very familiar with the concept of household recycling, which is basically bringing in whatever unused and leftover components, like a plastic bottle. You can squash it, put it in your bin. It gets collected by a garbage truck and sent to these recycle plants that would break it down and basically facilitate the components to build new structures like that. Now, the cool news is that our cells actually figured out to do this millions of years ago. So this is now the cell with the nucleus again. What happens inside the cell in this kind of analogy to cellular recycling is exactly that. There are components to the cell that can get damaged, such as the mitochondria. I already mentioned that to you. Other things can happen too. A cell can be infected by bacteria. You can imagine that that would be helpful to get rid of that before it takes over. This can be sent to these bins, which in a cell has a fancy name called autophagosomes. They are sort of double-bound trash bag, literally little vesicles that would encapsulate and basically sequester these components, send it with other basically modalities of the cells that can transport things from one side of the cell. This is by no means a small place, but basically transport it to another set of vesicles, in this case called lysosomes. They're bags that are really important to keep under kind of like a membrane in terms of keeping it captured, because they contain really high-end and very efficient enzymes that can help, just like in the inside of our intestine, and you know how well decorated it is because it's a very acidic compartment that helps digest components. So that's what happens when you then send it here. You can degrade these components of these trash bags that again can help build new and better components. So that's all good, except when autophagy does not work. So imagine that for whatever reason you run out of your bags or the vesicle components that would help capture the trash. What would effectively happen is, if you don't clean up your room or whatever, you're going to accumulate the waste. So that's what can happen inside a cell, and that's one of the things we're very curious to study. So what has been found in humans, at least as a growing field, is that, like I already mentioned, how young healthy cells over years would accumulate waste. And so what's really been interesting to learn is that it seems to correlate in this case with the fact that the cellular recycling, or autophagy, which runs very nicely in young cells and helps keeping it healthy and clean, tends to fail over time, and that correlates with this accumulation of the cellular waste. Now, this is also true in worms. We and ours and other labs have shown this to be the case. And of course, I'm just going to highlight here again, the real key is that we can study this only in the matter of a few weeks. So there as well, a young cell over time will become an older and cell containing waste or different damaged components, like misfolded proteins, or like these leaky mitochondria that no longer can function to be these energy factories that they normally do. So that's all bad, as you can imagine. The cool thing that we and others have found, and that we're really excited to study, is that these long-lived mutants somehow have figured out a way to keep this young, healthy cell in a state for much, much longer. And what's true, and we have no exceptions to date of the many different kinds of ways we can make animals live longer, is that they all seem to have found a way to keep autophagy running, and so keep their basically refrigerators clean, or at least not have very much waste in them. So we're really curious to study them, right? To figure out, so how do you do that? How do you keep your cells so healthy and also less prone to disease? Some diseases, for instance, relates to accumulation of basically clump protein, as you probably have heard, like Alzheimer's disease, or clump proteins specifically in neurons of the brain. And so again, if you could imagine you can boost this or keep it boosted in a way, that would be helpful for keeping those kind of stave off some of these problems that accumulates over time. So that's really a main objective for our labs, to learn more about, how is this process regulated? How do these long-lived animals figure out to like keep these trash bins going and have really efficient trucks to pick them up, and efficient plants, to turn over whatever it is that seems to be a good thing to turn over. And obviously studying this in a multicellular organism that have different tissue would also engage us in terms of asking, where does this happen? Is there tissues of the animals where this is particularly important? I mentioned neurons, right? That are not cells that can divide themselves and in a way get rid of waste by simply diluting it. So there you can imagine that something like a cleanup process like this could be even more important. So these are sort of our key questions. I'm going to just highlight one specific avenue that we're trying to take. We study in particular these, I mentioned, dietary-restricted worms. They are worms that effectively eat a little bit less. They live longer, they are healthier, and they also have less disease, like I mentioned, as opposed to the short-lived normal worm. So the normal worm has a number, and this is something we're starting to learn, of different types of bins, if you wish, to put different types of trash in. I mentioned mitochondria, or it could be lipids, or it could be these clump protein that normally runs, as I mentioned, well in a younger animal. But we're interested in learning what happens when you're now this long-lived animal. So what we're finding, but we're still investigating, is that it seems like these longer-lived animals, they have many more and probably a bigger variety of ways to put away their waste. So they're basically, their trash system seems to be much, much more efficient. And so by having seen that they have more and they seem to work better for overall turnover, it's of course this idea that we're now exploring, if there are ways that we could maybe engineer or boost, either with drugs, so we can do it by other means, by different regimens, but we can make either the trash bin bigger, or we can have more of them, and that somehow could help these animals also do more recycling better. And so that, of course, would then prompt the exciting idea that maybe this could give a longer and also importantly healthier life. So I want to just highlight a real publication that we published December last year, where we actually did manage to kind of express or put in more of a certain type of bin, a protein called P62. It's a receptor for some of these clump proteins. And if we give more of that, it seemed like they were actually much better at forming the trash bags and sending them to this recycle facility. So we could show that gave worms a longer and healthier lifespan. So we're really excited to learn more about each of these different types of bins and trucks and how we can manipulate them in a healthy way. So that with that, I'm going to just try and summarize what I have told you today. I have shown you, or at least given you some snippets of how there are many genes and processes that can extend worm lifespan. I mentioned this insulin mutant, the insulin receptor. There's ways to do dietary restriction that also extends lifespan. There are ways to give stressors to animals. So think of what doesn't kill you will make you stronger. If you, for instance, give the worm a little bit of heat, not too much, but a little bit of heat, you can come back actually and challenge them with more heat later, and they would be more prone to basically stave off this stress. And other ways we can manipulate the reproductive system or even mitochondrial function, these energy factories, like I mentioned. In all of those cases, and I also told you, we have really no exceptions so far. It's probably not going to be the whole way that we think of how you can extend lifespan. Other processes are happening too, like general metabolism I mentioned. But it's true that for all of them, they seem to at least in part engage this way of engaging cellular recycling to their benefit in a very beneficial way that we're trying to investigate. I didn't have time to give you all the proofs. This is not work by us, but we like to think that this is actually really of great importance, because similar links have been made in yeast that are also long-lived, in flies that are long-lived, or in mice that are long-lived. And this is really an important way to think about how the field has gone from these 30 years ago. I mentioned that this insulin mutant was shown to live longer. This idea that one could manipulate or change a gene and this would in itself affect lifespan, I think was seminal for thinking about lifespan or aging as a phenotype that was sort of plastic and could be maybe modulated in a way that we could think of ways to combat maybe even age-related diseases, or even possibly maybe one day reverse some of those aspects. So that's really how the field has now gone, to look in a number of animals to find that there are many different genes and ways to do this, and they seem to be highly conserved, which again gives us good indication, or at least premise for thinking about ways to interact with these mechanisms also in humans. I just wanted to mention these are some of the avenues, again, we're taking to study it in the similar organism, but with the real goal, as I mentioned, to think about how this would affect humans, us, in terms of living a healthier life for longer, and possibly also, like I mentioned, work as a way to prevent or even reverse disease. Like I said, these are not places where we are. There are efforts to this idea of doing geroscience, so studying gerontology in humans, not just for the phenotypes, but really understanding the genetics and how we could possibly intervene. For now, there is not, many direct ways, except some real ways that involves lifestyle choices. So I'm sure you've all come across this, that for instance, better exercise will give you a better health. And actually what's interesting to note here is that exercise, there are some data also now in humans, but definitely some really exciting data from mammals that shows that exercise in itself also tends to increase autophagy, this idea of getting rid of unwanted components that again could help you increase sort of the health span. So these are very exciting developments, something we're also interested in modeling in worms. We can swim them in liquid and sort of make them go on a treadmill in that way, and just in the same way try to learn and understand the molecular mechanisms. Ultimately, who knows if this would also come with the benefit of a longer lifespan. I leave that up to every one of us to think of that's necessarily the goal we want, aside from definitely living a healthier life. So I hope that our research one day will take us somewhere along this wellness road, I guess. With that, I'm going to close and just acknowledge the people that do all this work. This was a pre-COVID time for a set of bubble ball. This is how we interact now in the time of COVID. I want to especially acknowledge Jose Nieto Torres, a postdoctoral researcher, and Ify Tan, also a postdoctoral researcher in the lab, who has greatly helped with some of the cool slides. Jose himself has actually been a former speaker in the Fleet seminar series as well. So with that, I am also just going to acknowledge our funding sources. None of this would be possible without the generous support for our research. And I'll just finish on this one with just another slide for highlighting our institute. Feel free to join us in our mission. We are working in many, many different ways. We specifically on aging, but others work on other different kinds of diseases that we're hoping to find cures for. So with that, I am happy to take questions. Thank you very much for your attention.
Thank you so much, Marlene. I really appreciate it. I'm going to stop your screen sharing if you don't mind.
Sure.
But I'll make sure that I put the SVP website in the chat so that we remind our visitors, our attendees tonight to visit you guys, because like I said at the beginning, SVP does a lot of amazing work and they deserve all the support they can get. So we have a question. Yeah, go ahead, Marlene. Did you want to say something?
No, am I still sharing my screen or not?
No.
Okay.
I stopped your screen sharing. All right, so we have a question. Richie is wondering, what is the relationship between the DEF2 insulin receptor mutation and the insulin resistance seen in type 2 diabetes?
Yeah, so that's a really good question and a bit of a paradox, if you wish, to the field, because obviously, and this is true, Cynthia Kenyon told us these stories when she originally started working with this back in the late 80s and would go to seminars and there would be doctors treating diabetes patients and they would be like, there is nothing healthy about my diabetes patient, back at the hospital. This may just be a worm thing. And like I said, it was only later that work has been done in other organisms to show similar links. So I think part of this paradox had been basically realized by some of the later work in mice. And the point being that it kind of matters where you modulate this insulin receptor. So if you regulate the insulin receptor in the pancreas, it is a bad thing. And this mice that would have, had this kind of perturbation is not a long-lived mouse. And it has many phenotypes that resemble like a diabetic patient. But if you modulate it in the brain, for instance, this is when you have these benefits seen in the mouse, for instance. So we are just also starting to learn a lot more about these tissue-specific effects. I think a lot of the answer is to be found within the different tissues. But it's an excellent question, and it bothered a lot of people to that end. And actually, I think the field for a long time was dismissed as a wormy idea because it didn't quite make sense. But with the newer results that shows more context-specific effects, and obviously that would just beg for a lot more research. When are we going to do this, and how are we going to do this? You don't just now give a drug, right, that would impair the insulin receptor. If you have it work in the wrong place, you could have all sorts of other problems, right? So that's a very important lesson that was also learned for the field in general. So yeah, so excellent question. I hope that answers the question.
It did for me, because I had a similar question as Richie did. And I was intrigued by that. Yeah. As we're waiting for people to type the next questions into the Q&A, I'm going to ask you some if you don't mind. So you mentioned that dietary restriction and dietary habits are a big factor of it. I have the feeling that research in that area is kind of lacking. We have so many diet fads and things coming up, and then you do this, and the next months do that, and don't do this, don't do that. What is your take on the research field in that, and what could people do to maybe educate themselves a little bit better on it?
Right. Yeah, so it is a big and huge field for sure. It is obviously very attractive from the sense that it's something you can do, right? And it doesn't involve something sophisticated as cutting DNA or changing genetic material or things like that. So we don't do this research. I work and talk to researchers that do it. They say that, so there's data in that field. A lot of it is compromised by the fact that humans that are like put on different types of diets are not good at self-reporting. So a lot of the studies that are set up to actually be able to control for a lot of this and learn some of these questions like the composition, right, the timing, are not always powered enough to make sort of solid conclusions. So I think often then it ends up being that the public gets sort of one answer and then another answer because another, like study would now come out that have been better powered. So I think there is a number of indications. I think the general notion is that, and this is again mostly work from the simpler organisms, that it's going to matter not only the amount but also the composition. Now, exactly what that composition is, I don't think we know, certainly not from a human perspective. There's a lot of reports now, and I think that's maybe the easiest paradigm to do. I haven't done it myself, but intermittent fasting, where you basically starve for a longer period of time, which may be kind of more like humans did originally, right? When we didn't just have a meal set on our table every 3 times a day, almost at the same time, right? Like for that purpose, I know that a lot of people feel that that's been greatly helpful, and we can mimic that also in model organisms to show that it will have similar effect. It engages autophagy. It does start to clean up, and get some of the benefits to that end. So I think that has a lot to do with lifestyle, right? So this idea of really nailing it for how and what would work for you. For me, I do a lot of sports. It's extremely hard for me to not eat, for instance, in the morning and do this sort of long, let's say, haul of what people would typically do, like 16 hours, and then only eat or eat as much as they would want the rest of the 8 hours. A lot of people would practice that. So I think it's going to become very personal, like how you could envision doing that.
Maybe personal medicine will play a bigger role in that, where we can look at the exact microbiome for the gut and genes and cells, and maybe
and there's
customize
precision medicine that would come into play to that end as well, right? One drug may not work for all, because all of these, I think of these different ways. I had this big scheme of the many different genes that may affect. There may be sort of all like little dials that are set a little differently in each and every one of us. So you can't just throw, a drug after one dial if that dial is already very low in your body, right? So you're going to have to learn more about how each of these different settings can be modulated, basically.
Yeah. And another question I had, you said that a lot of this study is being done in animals and not in humans yet. When do researchers decide it's safe enough to go into human studies? And for your research particularly, when do you see that ever happening, going into human studies?
Right. So this has been a long discussion. Like I said, it's only very, very recently that some of these human trials have been engaged. I don't know. So we can all, I'm sure, agree that it's a good thing to find drugs that would work against cancer as an age-related disease. I think it's a little more maybe controversial to think about addressing aging, right, as itself. I don't think of aging as a disease, but it may be sort of the stepping stone. It's a little bit like, do you get sick because you get old, or do you get old because you get sick? But I think with the data and with all the research that's going, there's a greater notion, right, that there are these common mechanisms that would allow us to maybe think of ways to intervene in, of course, a safe and sound way. But it has taken a lot of research, right? You don't just go do that the next day when you see a worm live longer, right? For very, very good reasons. So I think it's just been a natural development of these 30 years or 3 decades of a lot of research that, I obviously have not given any full justice to, right? With the thousands of people that would work in this field, but that collectively actually indicates some very strong leads that now would convince somebody like, for good reasons, FDA approved drugs and test those in a legitimate and in a controlled setting. So research is slow, but for many good reasons as well.
Yeah, but that brings us to a very interesting ethical question, right? Should we all strive to live longer and healthier, given the state that we find ourselves currently in, with limited resources? They're dwindling. We're having climate change issues, mainly caused by humans. So how do researchers address these sort of bigger ethical questions?
Right. Yeah, and I would emphasize, this is my personal opinion, as a researcher, our research does not address that, right? It addresses the possibility of doing that. I would say maybe my opinion has changed a little bit as I'm growing older myself, right? Like I have been in the field as of over 20 years where I myself have started to, get age-related phenotypes. And my personal opinion is if there are ways that I can improve healthier living, and by that I especially care about my ability to move. So, like to do sports, for instance, or to use my brain to, think about cool things or do research and help humanity to that end. These are things I would really like to work towards. Now, like I sort of mentioned in the end, does it come with longer lifespan? Personally, I that wouldn't be my, I'm not looking for the fountain of youth for that sake. But if it comes with that, like for instance, you could debate whether a cancer drug would extend lifespan. There's actually been some calculations, there are statistical, basically algorithms that are run. It's not necessarily clear because you may then imagine that another age-related diseases may actually take more prone, right? Like just like for the sake of fixing a lot of hygienic problems, like over this, whole century, we now only started to learn that you could die from more age-related diseases. Yeah. So I'm not sure it would cause like a big problem. It is true that because of the baby boomers and a lot of these people are now growing older, there is a real need to kind of understand this and help them from these age-related diseases. But from a society point of view, I agree that this comes with ethical considerations that we all should consider.
Yeah, and we have a question. How do we extend the healthy lifespan at the time we're in our 20s? I don't want to be a toddler or a centenarian forever.
Right. Yeah, excellent point too, right? So that was sort of the point I was trying to allude to, that we're very interested in understanding, where and when, right? Because again, too much of a good thing may not be so good after all, right? One example was the rapamycin, this drug that I mentioned, FDA-approved drug that's been shown to extend lifespan of mice. Actually, in the beginning, there was a problem with the formula of how to deliver it to the mice. So it turned out that the mice really only experience more rapamycin much later in life, which was an important finding too, right? Because it would be important to be able to do something once you have, you realize you may have a propensity for a given disease or something like that. So I would never modulate these growth-related processes, like when you were in development, or like, let's say your early 20s, that can be a big problem as well. We know that, and even from the perspective again of cellular recycling, it had its own roles during development, where you can imagine it's important for reorganizing, the different reconstructions, cellular differentiation. There's a lot of things that have to happen during that period that may be very different in terms of what trash bin you want to use when you're 20 from the trash bin you use later. So I think the timing perspective, it's a really, really important point. So the question is well taken, that we need to really be mindful of when we can think of doing things in order to achieve what we would maybe like to achieve.
Yeah. Can you walk us through a little bit? I'm kind of still interested about the ethics part. How do scientists approach the ethic? I mean, is part of your PhD training are there ethics classes? Is there every time you approach a new research topic, do you have to answer these questions? How does that all work?
Well, I mean, so we certainly have ethics courses and, responsible conduct of research. I would say that's more from a practical point of view, like how do you conduct your research in a respectable way, like when you especially work with older animals, right? Like mice, we don't ourselves work with mice, but you would have to have an animal protocol and plan in place, right? For how you would take care of the animals, especially older animals. But I would say the more sort of philosophical, ethical questions, no, to my knowledge, this is not something we teach, but we talk about, and we should keep talking about that, because as you know, sometimes actually research probably moves faster than even sometimes the law and regulation does, right?
Yes.
And so it may be possible to do things, quicker than we've actually thought about the real ramifications for it. So I'm definitely for that. But I'm also cautious in the sense that for the sake of aging research, there's a lot of research still to be done to hopefully harness the best aspects of it for mainly, in my view, the disease aspects and how we can, provide better cures and better possibilities for humans that have age-related and other diseases.
Yeah, I agree. I agree that research oftentimes works a lot faster than the law or things can keep up. We've seen that in tech, right? I mean, it's just one of those things.
Right, exactly.
So since you've done the research, since you've been working in this field for a long time, what are the changes that you made personally in your life after realizing some of the things you've learned in research?
That's a good question. Well, I've always been very active, so this idea of especially exercise being, like a very, also a thing we can do, right? Aside from, let's say, considering how and when and how much to eat. I think exercise is especially important. I was especially excited to learn from now a recently deceased researcher, Dr. Beth Levine, was one of the first to investigate autophagy and its role in exercise. She herself, as an example, when learning that exercise, these mice induced autophagy and they seem to be healthier for longer, not only as better muscles, but actually also had better brain function. She herself went out and bought a treadmill that day to basically start running more. So I wouldn't necessarily say I changed so much behaviors as just being more aware of it. My former advisor, Dr. Cynthia Kenyon, in the early days, also when investigating the insulin pathway, which we know relates to sugar because insulin signaling relates to sugar especially, when we gave more sugar to the worms, that was actually something that would make them shorter lived, consistent with reduced insulin, making them longer lived. And at that point, she cut sugar from her diet, actually. I think I live a sort of more moderate, maybe sort of like the old Greek, the middle way is going to be, a good way. But I do take note to exercise and eat well and keep that sort of mindful. I don't take any of the drugs that are available. I know other of my colleagues, may do so or practice intermittent fasting as another way. And maybe I'll try it one day if I'm not, like if I get injured or something, then I could test it out. People say over a little bit of time, it will actually provide some benefits and, you sleep better, you feel better. These monkeys, actually, that was one of the things, obviously, with more complex organisms, you can measure more traits, right? So, their ability to sleep and do different tasks and smell, it seemed like a lot of phenotypes were basically improved to that end. Yeah, so very, very exciting, but early days to say whether it would really help for humans in that regard.
Wonderful. Thank you for sharing that. I just made a poll available. If people could take just a minute or two actually to fill out this poll, it helps us gauge how you enjoyed the event, what we might change to make it better for the next one. So if everybody could take a minute, that would be great. And I'm going to ask you another question while people take a moment. I had wanted to do that poll earlier, but I completely forgot. That's the thing when you have to multitask on a call. Sometimes you just forget. So what's the next big thing for you? What are you hoping to see with your research within the next, like, say, 5 years?
Yeah, so I think the field of aging is a very exciting time, like many other research fields, just because technology is at this point so much more advanced that we can basically modulate in a much more detailed ways, both to the questions that came up earlier with sort of time and place. I think it will be important to learn more about how these garbage trucks and the bins like kind of work together in a way that we can hopefully induce the benefits of this autophagy process. and not have other like side effects. So many of these long-lived mutants that I mentioned, including animals that are dietary restricted, it, for them actually comes at a cost. So, which I didn't mention to that end, but many of them, for instance, would have reduced reproduction, so fewer progeny, right? A lot of it in that regard would be maybe sort of think of as a trade-off. I think it would be very important to learn in the field if we can sort of harness even this way of thinking about stress responses, these self-protective responses that are normally there simply because a cell or you and me, we would encounter stress just by living.
Yeah.
So to what extent we can really harness those processes, right, to kind of gain the benefits of it and really understand how they would work together in a disease concept. I think especially this concept that I also mentioned, that cells will tell each other like what's going on. So this intracellular signaling is a rapidly moving field, also again permitted by the ways that we now can modulate a gene in one tissue and ask, you know, for effects on a distal tissue. So learning about these intertissue signaling components as a part of aging, not just the things that happen inside the single cell, but how does the rest of the body sort of come along and either, you know, get a good signal or maybe a bad signal, I think is a very important part of, again, geroscience or this sort of genetics of aging, if you wish, and hopefully that would lead us into new areas of biology we never really knew. In fact, the latest area that is kind of coming along is that these proteins that seem to be important for autophagy and doing recycling is that lo and behold, they also sometimes tend to moonlight in other processes. So we're now starting, like for instance, secretion again of molecules that can help tell other cells what to do. So, you know, it's likely to be much more complex. And how that, for instance, would play into aging has nobody has looked at that yet. So that's something we're especially excited about, to think of, you know, non-canonical functions for these autophagy proteins and mechanisms.
Interesting. That brings us to a question that we just got. Is heat stress to extend lifespan translatable to humans? That'd be nice as we get into the summer heat for San Diego.
Excellent question. Yeah, so there are no data really to prove that, but what the example I like to highlight, also being a Scandinavian, is that our fellow Finnish people who loves to go to saunas,
yeah.
Actually, there is quite a few data and results and publications to show that these, and you know, you can control for all the other things that Finnish people do, you know, like shoot vodka or whatever else. But there are data actually in a control way that can speak to that they have reduced cardiac issues, for instance, and other benefits. So I don't think it's sort of far-fetched to think about it. I would also emphasize that exercise, that's well known, actually also works by increasing temperature. So whether there is sort of a way to, again, in a good way, not too much of a, you know, this good thing to take it too far. So if we can understand these self-protective mechanisms enough, for instance, like heat exposure, so for people that go do maybe hot yoga, right? So you both have to move and you also have to get warm, could be, you know, one new idea for a new study. I don't know if anybody has done or think of doing a study like that, but that's would definitely be a way to think of a translatable idea, right? Like that exercise and temperature could help that way. So yeah, some indirect data, not exactly to aging per se, but definitely to some of these age-related pathologies like cardiac disease would be.
Interesting.
Yeah.
Hygge helps too, somebody says.
Say again?
Hygge helps too, somebody says.
Yeah.
That's one thing I miss from Germany, all the sauna spaces they have.
Right.
I do feel better every time I can go. Doesn't happen as often here, but yeah.
But obviously, if you give too much heat, that's not good, right? Then you're gonna feel exhausted, and so it's sort of finding the sweet spot and understanding what the true meaning of like that state is to the organism.
Perfect. And we've come up time. We actually went a little bit over, so thank you for your extra time. I really appreciate it.
You're very welcome. It was a pleasure. I enjoyed it very much. Thank you for having me.
Wonderful. Thank you so much, Malene. And then
Bye.
thank you everybody for joining us tonight. I hope you will join us again for future talks. We will have more talks with SVP and with other institutions here in San Diego to feature more scientists that do amazing work in San Diego. Please be sure to visit the Fleet website as well as the svpdiscovery.org website to follow us on all the things we do. Follow us on social media. Check us out on YouTube. We have plenty videos and science engagements for you there as well. And with that, I want to wish you good night. Stay safe, stay healthy, and I hope to see you soon. Bye.